All right, hi guys. So welcome. We are on our next video for genetics. We are using the book Concepts of Genetics. And if you go back and if you have seen the introduction to genetics video, you saw the timeline. And if you haven't, you can go back and watch that. But looking at the timeline, we still haven't discovered in this point in our studies, we haven't discovered the makeup. We haven't really discovered what DNA is. It's at this point that we've recognized, like, hey, chromosomes they have the genes that will express the phenotypes. And so as we're going through today, we are going to be talking about sex differentiation, and we are going to be talking about, or sex determination and differentiation. We are going to be talking about chromosomal mutations. We're going to be talking about crossing over and genetic recombination and how that happens in a eukaryote versus a prokaryote or bacteria. And then we are going to be talking about extracellular DNA. So we have a pretty big roadmap. As a matter of fact, in your book that covers, I think, like 4 chapters or maybe even 5 chapters. So it's a big chunk, but what I found is the way that it can flow together, it flows really well this way. So don't panic, don't freak out. But remembering that to make information stick, there needs to be some action for the nervous system. So you need to be writing something down or listening and then listening again and writing it down so that you start to create pathways, so that you're not just cramming in information and then poof, it's gone the next time. Now, some of you might just want to do that, whatever, but I don't recommend it, okay, as the teacher, okay? So we're going to start with sex determination. Now, in humans, we know that sex determination is determined by that if you're XX, you're a female. If you're XY, you are a male. Hopefully this isn't like, I'm going to flip it for you guys. I don't want this to be bumping up against my chest and making weird noises for you guys. Okay, so we know that for humans, XY, Y determines that you're a male. XX, female. Whereas with flies, it's like the ratio of X's. What is it? Oh, my brain just went blank as to what birds. Birds, the female has the heterozygous trait. So it's like ZW, I think, for female birds. And then for crocodiles, their sex determination is determined actually by the environment. So they can't, like, if the temperature is extreme cold or extreme hot, you're going to end up with more females. If the temperature is intermediate, you're going to end up with more males. So sex determination, it's across the board important. And what they found is, like, if we look back at more ancient genetics, the Y gene was always there, or the Y chromosome was always there. And the Y chromosome holds the SRY gene, and that is the gene that encodes for the ripple effect of the testes to start producing testosterone and anti-Müllerian hormone, which will start to develop the sex characteristics of a male. So, long story short of your giant chapter, there you go. But we're going to be we'll talk about it in a little bit more depth. So determination is the chromosome and the gene. Differentiation is hormones like testosterone and anti-Müllerian hormone. Okay, so we know determination is genetic, differentiation is hormonal. Okay, just want to make sure and clarify those two differences. Okay, so the actual definition of sex determination is the biological process by which any organism develops as a male or a female. Okay, and so we're going to, of course, be talking about some terminology. And today, some terms that I personally, as I was on this journey, got a little bit confused about were gametes. And maybe some of you are like, gametes, that's such an easy concept. But, for some of us out there, it wasn't. But for me, it was not easy. Zygotes. Okay, that was a difficult one. Haploid. Okay. Diploid. We got germ cells. And we've got somatic cells. Okay, and so there's more that we're going to talk about, but the makeup of a chromosome. This is a chromosome, but so is this, right? Chromosomes are determined by their centromeres, okay? Their centromeres. And so just cleaning up some things that tend to get missed in this topic, and it is why this topic becomes kind of scary, is because there's so many terms that it's like they all sound the same, and like that is a chromosome and that is a chromosome. I'm getting confused. I don't know the difference. Okay, so we're going to dive in today, and we are going to go a little bit back to mitosis and meiosis, so that you can we're not going to go through each phase individually, but we are going to look at when they happen, okay? So we got to go back to fertilization, right? So when we are at fertilization, let's draw, and they aren't going to be to size so that I can make this happen. Okay, so we have dad. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. And then he can either have an X or a Y floating in there. Okay, so this is his. And this is a haploid cell. So it looks like this. Okay, in. Now, if we're going to look at mom, let's do mom in a different color. Let's do mom in this pink. There we go, pink. Okay, let's do the egg cell, the ova, like this. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, and she will always give an X. So that's in the ova, that's in the sperm. Okay, remembering that females are born with all the eggs that they will have. Males, spermatogenesis happens throughout lifetime. Okay, so right now we have 2, and so this would look like this as well. These are 2 haploid cells. Okay, these are gametes. Okay, this is dad's gamete and this is mom's gamete. Okay, now what's going to happen is they're going to fuse. Okay, they're going to fuse and they're going to become one cell. So let's make that cell a different color. We'll make it black. So it fuses right here. And let me get my colors, because I think color coding this makes it really helpful. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. And dad came in with a Y, right? He came in, he donated that Y here. Okay, so we know that Y contains the SRY gene, which means that this cell is going to develop as a male. Okay, now mom gives her 22 chromosomes. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. Okay, so we have 22, right? 22 pairs of these chromosomes, and they're homologous chromosomes. So the short ones are with the short ones, the long ones are with the long ones, and they each have different alleles. They code for the same thing, but the code might be a little bit different for each. Like the code might be green from mom and the code might be brown from dad, which we talked about last time. Okay, so now this cell is now termed a zygote. Okay, and now look at it, and mom donated her, let me put this here. Mom donated her X. Okay, so if we look at this, we've covered gametes. These are the gametes. Here's the zygote. Now what's going to happen here is this cell is going to start dividing. 1 will become 2, will become 4, will become 8, will become 16, and prime numbers start to get too big for me to go through that, but we're developing. So we're becoming, and this process is happening through mitosis, right? Mitosis. So let me give you a little example. So if we look at this, we have 2 cells that came, the first 2 cells. They're diploids, right? This zygote is now a diploid. So 2 in, they have both. This one is just in, right? Okay? I don't think that 2 is supposed to be in there though. I think it's like this, 2. Okay? All right, so, and then as these split and become their 2, right, because as mitosis splits, you're going to get 2, right? There are 2 in, 2 in, so then you have 4. So you see the process. All of this is the same as this beginning one as it goes. Sorry. Then as you're getting all of these trillions and trillions and trillions of cells that make you up, they're all diploid cells. These are your somatic cells. Okay, these are somatic cells. And these cells are going to start to differentiate, and there's a lot of embryology involved here, and that's a big topic in and of itself. But some of these cells, they'll become my brain cells, my neurons, right? Some of these cells are going to become my digestive tract. Some of these cells are going to become my eyes. You know, you get what I'm saying? So these cells are going to start differentiating to be the different parts that make up me. And some of them are going to differentiate and they are going to become my my germ cell, like they'll be the germ producing, or the germ cells, or the germ organs. My brain is not remembering specifically what that's called. But your sex organs, your sexual organs. So all of them start to differentiate and you become you. So remembering, oh, the gonads, there's the word. I knew it was a G, but it was just not coming to me. Gonads. So the female gonads are the ovaries. The males are the testes, right? And so they're going to differentiate, and they differentiate through, or they split and they divide through mitosis. Differentiation, some of the genes will be permanently turned off. So once these cells become your liver cells, they don't really turn back on and become your heart cells. Like they can't differentiate to do what your heart does. Your heart cells do a certain thing, your liver cells do a certain thing. They don't really cross over and do what each other does, okay? Here we go. So somatic cells. And then, so once they become gonads, then you have those cells, I'm going to use this color again, those will become your germ cells. Those gonads will start to create your germ cells, okay? And these are your germ cells right here. Your sperm, your ova. Remembering that this is already, you already have the eggs. They just need to be matured, okay? So now when we are going and we are starting to create these germ cells from the gonads, that process happens through meiosis, okay? Now, remembering that mitosis has PMAT, that's what you can remember. Prophase, metaphase, anaphase, telophase. And then at the beginning, there's always interphase where all of this is copied, duplicated, but you still, in mitosis, will end up with 2 new diploid cells. Whereas in meiosis, you have PMAT 1 and you have PMAT 2. And this is because you're getting copies from both parents and then you're splitting it up because you don't want to have, so say we had all the pairs. So say it looks a little bit more like this up in here and a little bit more like this. And so when they fused, what would happen is you would have an overdose of chromosomes, okay? You would have an overdose of chromosomes, and that is not something that you want to have happen. So there's an important thing that's happening in meiosis. So once those chromosomes have duplicated, right, and they duplicate in interphase, let's erase this right here. They duplicate in interphase, and so you'll have your, I'm going to draw this up really quick. Let me just get her out of my face. Okay. All right. So now you have right here, and for simplicity's sake, I'm going to divide them up. So you'll have mom 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 15, 16, 17, 18, 19, 20, 21, 22. Same thing for your dad. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 19, 20, 21, 22. And then we have the Y and the X, right? Okay. So this is mom's side, maternal, and this is the paternal side. So they've all duplicated. Okay, this is happening in meiosis. So we're going to take this set right here. Okay, and they're going to do, and honestly, they are mixed up. They aren't automatically just split up like this, but this is just for simplicity's sake. So we're going to take these two, and this happens with all of them. They're going to have like a little dance. Okay, they're going to do like a little dance of a crossover. So we don't, I want to keep that just to be safe, but we'll take this out of the way. Okay. So we have these two cells, and I'm going to draw them kind of big so that you guys can see what happens. These are our chromosomes, right? These are chromosomes, individual ones, homologous pairs. These are also considered chromosomes, even though they're individual. Remember, it's the centromeres that make them a chromosome. Just wanted to clarify that. So we have a chromosome from dad and a chromosome from mom. And what's going to start happening is they're going to start dancing, right? They're going to start getting closer to each other, moving closer to each other. And I'm going to make dad move closer. And what they're going to have happen is they'll start crossing over. Boop. Let's draw this like this. Okay? They'll start crossing over, and maybe they cross over right there. Okay? But there's multiple ways that they can cross over. But when they cross over, there's a little line here that will help them split. And then when they do this crossing over, what will happen when they come apart is it will look a little bit like this. This one will have like this little blue right here. And this one over here, will have this little pink region right there. And maybe down here, we could say the same thing. Okay? So this is called crossing over or genetic recombination. Now, let me also clarify something. This is one sister chromatid right here. This is the other sister chromatid. They're the exact same. They have the exact same information, right? And so if we just duplicated like this, it would be the exact same. But because we're having crossing over or genetic recombination, it's recombining this. And now let's say they get split up. When they get split up and they become the 4 germ cells, or the 4 gametes, it would look like this. So by the time meiosis is over, this one would go here, this one would go here, this one would go here. So let's look at this, the cells. So we would have this one here, and it would look like this with its blue. And then you'd have one right here, and it has its pink on the top. Okay, and then it's the same. Okay, so now we have a new combination of genes, right? We have a new combination. So these are the sex cells. Now remembering, with females, there is one female cell that gets most of the organelles, most of the cytoplasm, because we don't need to waste our time with these. So we just get one big one. So we kind of get the bar bodies, or not the bar bodies. No, maybe. No. There is one oocyte that gets most of the information, most of the goodies is what my teacher used to call it. Whereas all the males, they come away with this. Now, this recombination process does not necessarily happen on this XY chromosome. Let's look at this. On the XY chromosome, they have pseudo-autosomes, which, by the way, this is also an autosome. These are autosomal cells. They can be called that. And these are sex cells, germ cells, okay? And these are the cells for life, or autosomal chromosomes right here, okay? These are heterozygotes, right? Or heterochrome, oh my gosh, the word. See, this is what I'm saying, guys, the words. They cross over. But let's look at this. This has this little region right here. This is where the SRY gene is. Okay? Now let's say we had that crossover. And remember, and I'll talk about the pseudo-autosomal because I kind of interrupted myself. So say they crossed over. So now this female, this chromosome has this little region here that was crossed over, and it also contains the SRY gene, okay? But now, this little guy over here, he has the Y gene, but, which is the trigger, but he does not have the SRY. So it just doesn't work that way. So what they have is little regions at the end. They have regions at the tips, and they're called pseudo-autosomal regions. And so they can cross from that region, but only from that region, so that that SRY gene does not get crossed over and mixed up. Okay? All right, let me make sure I have everything. Okay, so we talked about genetic recombination. So let's look at this. Let's talk about chromosome mapping. So this is where they can map out where genes crossed over, or if a gene's gonna get crossed over at all. So we're gonna pull back up our maternal, and we'll pull up our paternal, okay? And they're gonna have genes, okay? And remember, this is happening with all of them, not just two, but we're gonna give some examples. So here's one. I don't want that to be the same color, because, right, it's a different allele. It codes for the same thing. Maybe it codes for your hair color. Okay, your hair color, we'll put it as red. Okay, and we'll say these code for hair color. Okay, now let's say up here, we'll use this purple and this purple. They almost look the same, but they're not. Okay, guys, trying here. I don't have that many colors to work with. And then we have black and blue, we'll say. Black and our blue. Okay? So what chromosome mapping is determining if these genes get, like, did they get split apart, separated from each other. And when I say separated from each other, I don't mean these two chromosomes separated from each other. I mean the actual genetic information. So is this one going to get separated from this one? Is this one going to get separated from this one? And when you look at it, which one is more likely? So when we just saw that crossover, that dance that happened, which was more likely? These bottom ones to get crossed over? Because, they can get crossed over, like sister chromatids, they can actually cross over. And that would be coming from, like, that would be two strands. So say dad's crossed over here. So there's 3-strand double crossover is what it's called. But the sister chromatids, they can also cross over. Now, because they're holding the same information, it's irrelevant, but they can still cross over. So let's just say here, we're looking at this, and there's a term that I have to introduce here. It's called a centimorgan. So it's called a centimorgan. The guy Morgan, remember in our timeline we talked about him, he discovered this. So when we're looking at, it's a 1 to 100 ratio, or percentage, that it will cross over. So say right here, this distance, this is a distance, right? Distance between genes on the same chromosome. Okay, so here's a distance here. And we can say it's like 10% or 10 centimorgans. Whereas this right here, we can say is like 50. Okay, so which one is more likely to have happened? So if it's 1 to 100, we have 50 right here. So that's about a 50% chance that these two will get separated from each other. Whereas this one, they only have that tiny area to work with. They have a 10% chance of getting split apart from each other. And so as the space increases, they're one more likely to cross over or to get separated. And when I say crossover, remember we just talked about crossover or recombine with something else. If we were to look at this, the greater the space, the greater probability that they are going to get separated from each other. The greater the space, it's also the greater, we have more, what is the word I'm looking for? More mistakes when we're mapping. So the greater the space, the greater the probability that there will also be a mistake. Okay? Yeah, that's pretty much that example of that. So let me give you an example showing like the crossover that happens. So say these two, they cross over right here. So let's draw this with all of its dynamics. Okay? So we have here, whoop, and we'll say this one gets over here, so we don't have to reopen that pink one. And then here's the blue one. Boop. Boop. Okay, now this purple stayed the same, right? And this black stayed the same. But this red is what came over. So this changed. Okay? Then we look over here, we have this purple, it stayed the same. And this blue, wherever it went, stayed the same. But this orange crossed over. So you can see this genetic recombination. Genetic recombination happens from crossing over. Okay? That is in eukaryotes. That's what happens in eukaryotes, okay? So now this does not happen during mitosis, right? Mitosis, we don't want this to happen. Sorry guys, I don't want that hitting that. During mitosis, we do not want this crossing over to happen. We want it to stay like this. Now, if there is some type of mutation within mitosis, it is not going to get passed out through your genetic information. So it only, like that will only get passed on if it happened within the germ cells, okay? Or before, okay? If you already have that. Does that make sense? Hopefully that makes sense. So we've talked about that. So now what happens, this is in eukaryotes, right? And so let me just for safety, say this. Eukaryotes, remember, they are nucleated organisms. Eukaryote means true nucleus, okay, right? They have nucleus and they have membrane-bound organelles, whereas prokaryotes, they don't, right? They don't. So our genetic information, technically, this is all within a nucleus, right? It's all within a nucleus. Now, a prokaryote or a bacterial cell, we'll just stick with the word bacterial cell. Bacterial cells, they don't have a nucleus, so their genetic information is just floating around. It's just floating around in the open, in the cytoplasm, okay? So how, like if they are going to just reproduce in bacterial cells, this is called binary fission. Okay? But this genetic information doesn't change. So how do they switch up their genetic information, and why does it even matter? So say we have an antibiotic or something that can, antibiotic that can kill this one. Well, then it's going to kill every line of this. This brings up a fun point. Cheetahs in our world, I don't know where else they would be, but cheetahs, they are so genetically similar that scientists are actually concerned that if a disease were to hit cheetahs, that because they are so genetically similar, they won't be able to stop the disease from spreading through all of the cheetahs. So, like, you know that with humans, you can't just give a skin graft with somebody that doesn't have the same, like, your body will reject it if it is not matching for you. Whereas with cheetahs, they can just give each other skin. Like, it don't matter. Like, if a cheetah gets burned and somebody intervenes and they get some skin from a different cheetah and they put it on this cheetah, well, it won't reject it because they're so genetically similar. That's a fun little side note for you guys. Hopefully I didn't derail that. But that's the same thing that happens with bacteria, is if they just If they just keep going the way they're going and they don't have any diversity, one thing could wipe out the whole system, right? The whole, what's the word I'm looking for? The whole species. There you go. Kind of like the whole species of cheetahs. I'm going to keep that there just for safekeeping. Okay, so with bacteria, we're not going to go through like all the loopholes of this, okay, because microbiology will cover that. But we have 3 different ways that this can happen. Technically it's 4 ways, but we're going to cover 3 of the ways. So we have conjugation. And so the best way that I can think of this is like you have a secret note that you need to get to your friend. And you got that secret note in your hand and you shake hands with your friend and pass that secret message on. Okay, it's a vital secret message and you pass it on. So what with conjugation, I know that might sound weird, but hopefully you can remember that. Conjugation, shaking hands. So you have an F positive. So it has its genetic information floating out here. It's chromosomal information, which by the way, chromosomal information in bacteria is just a loop. It's a circle where ours is bound up when we're dividing. When we're dividing, it makes these chromosomes, these really tight structures. With bacteria, it's just in a circle. And in all actuality, in our cells, whenever we are not dividing, when we're in a growth stage, our DNA is not wound up. It's actually like that as well. Okay? It's once we start to divide that it becomes compact. Okay? Because we need to be using it, right? So we can't just keep it constantly packed up. It's zipped up. They don't know that yet in this time in history, but the actual chromatid it's spread out, and then this is when it becomes a chromosome. Okay, is when we start to divide. Hopefully that makes sense. So with our bacterial cells, we have our 2 bacterial cells here. Okay, this one is F positive, which means just basically it's fertile. It has genetic information. It's called a pilus here. It has genetic information, or well, this is called the pilus. It can create like a bridge or a tube. But it has this ring of genetic information that my brain is completely forgetting what it's called, but I'll put it in the comments. This ring of genetic information, or you can, if you know what it's called, it will pass from, it can create this bridge. This little piece of DNA, genetic material, can create this bridge. That's what this F positive means. This one is F negative. It does not have the ability to create this little bridge to pass this genetic information on. Okay? And so what it will do is it will make a copy, whoop, and then that copy will whoop, funnel over here, and now that one has that information. So this could be holding the information to create more pilus, more of these little bridges to tell more about this information. Or it could be, hey, this bit of information, that this gene that I'm sending over prevents a certain medication from being able to break through your cell wall. Okay? So that's the importance of genetic recombination there. Genetic recombination allows for diversity. Okay? Like I said, with the cheetahs, if one thing hit, would we be able to recover? As humans, yes. But as cheetahs, maybe not. Who knows? Transduction. Yeah, transduction. So transduction is like your Uber driver picked up somebody else. Like this wasn't meant for your Uber. And so this Uber driver is passing on other people. So it's passing on information. Another way that I think of it is, so this has to do with bacteriophages, which is basically just a virus. So you have bacterial cell, right? And it has its genetic information, right? It's chromosomes. This bacteriophage looks like a little alien, right? Comes in, and it has its own genetic information. Sticks it right in there. Now, what it does is it either sticks it in with that genetic information, splits up that genetic information, But what it's doing is it's creating a whole bunch of copies. And then what it's doing is it's creating more of these little phage bodies. Those look so beautiful, I know. They look all the same. But what's happening is some of them will have the old viral, some of them will have the new bacterial information, right? So we have all of this going on, and eventually this gets so big and it pops. So now you have all these little phages, all these little viruses, bacteriophages. My gosh, I can't draw when I'm talking. But it has this bacterial information, and it can go stick it into another bacteria with different information. And so it's passing on that information. So this just happens over and over again. Okay? The next one, so that's transduction. The next one is transformation. Okay, transformation is basically like you picked up a dropped card. What is business card? You picked up a dropped business card. So let's say we have this bacteria. He's harmful. His genetic information has some exotoxin or something in there. So he's a harmful one. Say this little guy right here, he's chill. He's non-harmful. Non-harmful. I don't even know if that's a word. But we're going to say it today. Let's say something comes and it kills this one, right? But now its genetic information is floating around, right? Because this just doesn't poof disappear, right? We got to break it down. Well, let's just say if this has the ability to do so, right? It has to have the ability, it's called competent. It has to be competent. It has to be competent. If it's competent, it can bring this information in, add it into its DNA, and now it has that harmful information. So it can do, maybe it can start producing that exotoxin or endotoxin, whatever that might be. But this is important for making sure that there is genetic diversity, right? Genetic diversity. The same with us. We have that crossing over, we have that recombination so that we have genetic diversity. It's the crossing over is for diversity. What happens is you think of it like it's nature's big experiment. Sometimes experiments go wrong, and that is where let me make sure I'm not getting too far ahead of myself. Mutations occur. Okay? But before we go on, there is one more thing that we need to talk about. We need to talk about, let me erase this too. We don't need this anymore. We probably did, but it's okay. So when we're looking here, and we are looking at extranuclear inheritance. So there are some organelles outside of the nucleus, right? So we have our cell here. We have our nucleus that has our DNA in it, right? It has our chromosomes in it. I'm not going to draw them all out again because I'm probably going to have to draw them again anyways. But we have organelles that have their own genetic information in them. And that would be the mitochondria. Okay? It's from the mitochondria. Now remembering, I just erased that, and let's just draw it again. I'm going to do it in the same color. So remembering that this oocyte has all the cytoplasm, all the organelles. This little sperm cell has its chromosomes and that X or that Y, right? It doesn't have a whole bunch of organelles with it. It's carrying information, basically. It's like the mailman, okay? This oocyte has its nucleus, right, with the genetic information in there, but it also has cytoplasm, it has organelles, right? It has all of the stuff to make a new person right there. So which one do you think is passing on? It has its DNA in the nucleus and it has its DNA within the mitochondria. So where do you think we get extranuclear information from? So this information that is from the mitochondria, and in plant cells it's in the chloroplast, right? It's in the chloroplast. And so we have mitochondrial DNA, and we have chloroplast DNA. And it's always coming from the mother, maternal. Because that maternal has the organelles within the cell. The sperm cell does not. Okay? So what they found was these, like, why is this a thing? Ancient, like if they were to look back on it, it's a hypothesis. What they're saying is this mitochondria and the chloroplast, these little factories, were once organelles that were outside of the cell. So this is a hypothesis that they had. And what they found was that what they think happened is that this mitochondria that was once an organelle all of its own, this chloroplast that was once an organelle all of its own, had a mutual agreement with this ancient type of cell. And it started working together, and they just kind of developed together, and this never went away. This genetic information never went away. Now, what kind of information does this genetic information carry? It's basically metabolic processes of these cells. So this is a really, these have high oxidative processes. So the genetic information that's there is from DNA. Now that's a hypothesis. Okay, that's just a hypothesis. I'm not saying I agree with it or I disagree with it, right? Evolution, things like that. They evolved together. That's what they're saying. They're two different organisms that evolved together. Now the proof of this was, what they found was with chloroplast, and this is actually anti-Mendelian, right? Because they're saying you get information from both mother and father equally. Well, this disproves that, because this is only maternal genetic information. This only comes from the mother. And what they found was, this guy doing the research, I can't remember his name right now, but he was looking at a certain tree, and what he found was all this stuff would happen. So this tree, I'm going to draw it here, and it's not going to look good, but so especially since I don't have any green, so some of the leaves would just be green. Some of the leaves would just be white. But some of them would be a little combination of the two. Okay? Green and white. And so what he found was, he did all the different ways. He did all the experiments. But what he found was that this was coming from the chloroplast. So one of them had a mutated gene for chlorophyll. That was the white, right? The white ones. And then one of them did not. So that was the green ones. Then what he found was, when it was a little combination of the two, when there was chloroplast with a little mixture of both, you would get both. Okay, so you would get both of that information. And that was only coming from the maternal. So he tried it from the paternal side, and it just, this did not occur. It only occurred from the mother side. So this is an anti-Mendelian effect right here, the process that happens. It goes against everything, like everything that Mendel, not everything, but that there is this segregation of both mother and father genetic information. Okay? So that's extranuclear inheritance. Now, let me make sure. We got crossover, we got chromosome mapping, we got extranuclear inheritance, we've got the difference between prokaryotes and eukaryotic genetic recombination, with conjugation is like the handshake with the sneaky note. We've got transduction, which is like an Uber driver who picked up a different Uber driver's information and is carrying it. It's not the best analogy, but it works. It has to do with bacteriophages, right? Recopying their viral information and passing on new bacterial information to a new bacteria. Okay? And then transformation, this was picking up a dropped business card, right? So or even better, picking up somebody else's $100 bill on the ground, right? Yay! And now it's yours. Now you have it. Okay, and you can pass it on if you want to somehow. Okay, so that is the difference. And with genetic recombination, it happens within meiosis in eukaryotic cells, okay? All right, so now we need to talk just a little bit more about sex determination. So we talked about the difference between determination and differentiation. Differentiation is those cells that are dividing in mitosis, they differentiate and they become gonads, right? And those gonads are male or female, and that will determine, they will split in that X and Y, that Y, SRY. I don't know why I keep saying SRY. SRI is a process I use with patients, but SRY gene will determine, and it will flick on the process of differentiation, sexual differentiation. So that development of testosterone from the testes and anti-Mullerian hormone. Mullerian hormone. Just making sure I'm saying that right. Okay, so the Y chromosome determines. It's the determinant in mammals, in humans specifically. This is not a 1-to-1 ratio, right? There's actually more males born than females if they were to look at this. Now, what happens, we have to talk about this, what happens if a female has 2 X's, right? We don't want a double dosage of this X information, right? So what the body will do through an enzymatic processes is it will do what's called the Lyon hypothesis. So lyonization, let me write that down for you guys. The Lyon hypothesis, or lyonization. And what that's doing is, you can think of it like a volume knob. It's an enzymatic process that shuts off one of these X's, and it's random. It doesn't matter which one. They'll just shut off one of them. Okay? It just shuts off one. It just becomes a little bar body. And when the one that's off is called a bar body. Okay? And then the other one has its information, right? Okay, and that's what makes the, remember we have X dominant or X recessive disorders or developments, mutations, okay? Erase all this stuff here at the top. We got that down. If there's any specific terms that you're not understanding, make sure and put those in the comments, because if you do that, then I can really see the holes, because I can't see the holes in your learning, like where you're not getting it. So put it in the comments or email me, message me, so that we can really get that cleared up. Okay, so now we talked about crossing over, genetic recombination. That is nature's way of experimenting and creating new things. But sometimes there are mutations that occur, and sometimes they're great, and sometimes they're not so great, right? Our eye colors are actually mutations, which most people know that, but blue and green eyes, those are not necessarily, they're mutations, but they're not necessarily the most adaptive eyesight. They don't deal with light as well as brown eyes do. So that's a mutation, but we don't say necessarily that it's bad or good. It's just we have blue eyes and green eyes now. Okay? All right, so mutations in chromosome numbers. That's numbers. That happens through non-disjunction. So when those are splitting apart, when they're getting split apart, maybe they don't get split apart. Or maybe they get split apart and they don't get another one. This is called, I'm going to make sure I spell this right, monosomy. Where we lose one. So this would be like X0. Didn't get another X, didn't get a Y. That would equal Turner syndrome. I don't think there's an apostrophe. Turner syndrome. Okay? Now, trisomy, which we hear about, that's pretty much the only one. Here's the thing. All of these that I'm about to talk about, they're called aneuploidies, which means not true. Euploidy means a true pair. Aneuploidy does not. Okay, it means not true pair. So obviously we don't have a pair. But here's the thing, most of these end with lethal alleles. So they don't make it all the way through. And they found this through genetic testing of like spontaneous abortions. They would look at the genes and they would see, oh, like this had a mutation, but it was lethal. It was a lethal mutation. Maybe it's because they don't have enough of the autosomal chromosomes for life, right? Whereas monosomy, like you saw, we get rid of an X, but they're usually sterile because we can't give away, like we don't have anything to potentially give away, right? So they're sterile. They are female, right, because they don't have the X, but you can see that this is a process that happens. This is probably one of the only one that I read within the text that survives a monosomy like that. Okay, then we have a trisomy. So, right, we have the 3 right here. So if we're looking at our 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, X, Y. Okay, so with trisomies, it looks like this. So there's the 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. 13 is one where it can tend to be a trisomy, so they get an extra one. But I think it's like it's an R, sorry, P-A-C-A or C-U-A. No, that doesn't look right. I can't pronounce the word, so the name of it's not coming to me. Trisomy 13, though. 13, okay? 14, 15, 16, 17, 18, that's Edwards, tends to have a trisomy. 19, 20, 21. 21 is the most common. It's the one that most people know, and that's Downs, which is trisomy 21. Okay? That is where we look for our trisomies. Now we have a polyploidy. Polyploidy is more than 2 complete haploid sets. So the sets are that we have more than 2 of these. This pretty much only happens in plants. And with plants, they thrive. They love it. So has more than 2 complete sets. So this is like a copy and paste button for them. It's like, oh, I liked that version of that plant. Let's just stick it over in another one. So, or that version of myself, I'm going to stick it over in another one, because you have autopolyploidy, which is the same species. You get more than 2 haploid sets from the same species. Alloploidy is different species, so it's hybridization. So thinking like wheat, you get more than 2 sets of 2 different species. And then endo, this is within certain cells. Endopolyploidy, this actually happens within the human body, whether it's placenta or the liver. So these are highly active cells. And so sometimes what will happen, which I didn't know this until just now, what will happen is the liver cells will duplicate their material, but they won't divide as cells. And it is because they are so active, they need to be constantly producing, they need to be constantly reading, right? So think of if you have more than 1 set, then you can read this and make more proteins, right? Because your liver makes your albumin, it makes your transport proteins. Your liver does that. And so, and it's detoxifying, your liver is doing so much for you. And so thinking if it has 2 copies of that, it can work much faster, right? Okay, we're understanding that. And that's a type of polyploidy. It's called endopolyploidy. Okay, and I'm going to read you this definition. Specific tissues replicate DNA without cell division. It's to increase productivity. This happens in highly active tissues. Okay, so remember the significance of multiple chromosomal numbers, or the change in chromosomal numbers or polyploidy, sorry. It is your copy and paste button. You get more, like copy and paste, more liver DNA, so we can do more. Copy paste, we like this species, let's send it over. Okay, Now let's talk about some other processes, that mutations that happen within chromosomes. So we're going to talk about deletion. So let's just see here. I'm going to just pull this up right here. Just restart that right here. I could just draw an X, but I had to be difficult that time. So what happens is you have deletion. So we'll put this here, deletion. So let's just say something breaks off. Right? And so what happens is you have to, you could have it at the terminal end, or you could have it in the middle, right? And then these come back together, right? And so now the homologous chromosome, when it comes to pair, well, it's like, well, hang on here, wait a second. So what will happen is it will create this little loop, cut out that DNA, that information, and whoop, to make it the same so that they can pair together, because they can't pair if they're not homologous, right? So there's terminal or there's intercalary. So terminal is at the end, intercalary is in the middle. Okay, so this results in a dosage imbalance. Okay, you can think of this as you're reading a book and you just randomly tear out a page. My son did that, as a matter of fact, to my genetics book. Just ripped out a random page. That's deletion. He deleted that page out of the book. Sometimes it works. Sometimes you just delete maybe some interons, some information that wasn't needed, and sometimes you delete something that's important. It's kind of a hit or a miss, right? Depends. Okay, one of the most famous disorders is Cri-du-chat. And there are some developmental delays, mental developmental delays, but the way that they cry sounds like a cat crying. And I'm not going to reproduce that sound. So, but Cri-du-chat is French for cry of the cat. Okay, and that happens in deletion. It's like Cri-du-chat 5p. So a section 5p. I think that's in the 5th chromosome. Okay. All right. So then you have duplication. Duplication, just as it said. So with deletion, we had some missing or some loss of genetic information. Duplication, we have repeated information. Okay, so this can be in tandem. So we can, so let's say it's A, B, C, D, E, F, G. Okay, and then this other one right next to it is A, B, C, D, E, F, G. These chromosomes are linked by their centromere. I'm saying that so that you guys know that I'm talking about a chromosome. Now let's just say a duplication. So in tandem, A, B, C, B, C, D, E, F, G. So that's in tandem, right? Right next to the original. Okay, you could also have displaced. So it moves to another area. A, B, C, then you would have it move like this area. Duplication moves to A, B, C, B, C, okay, D, or even another chromosome. It can move to another chromosome. E, F, G, D, E, F, G. Now again, you can see that one becomes longer than the other, right? This is longer. The other one is reversed. So we could just flip that around. Okay, so the genetic information is not lost. So it's, but there's still unequal crossing over. So we do create that same loop where that our homologous chromosome that comes to pair, it's like, that's a little weird. We can't fit there. So whatever information is on that one, it's going to loop out, and an enzyme is going to cut it off, and we're going to get rid of it. So we do have genetic information that gets cut out in the other loop, but because we duplicated it, it's right there. Okay. But we increase the dosage of that amount, and there's an unequal crossing over. Right? So this is kind of nature's way of experimenting. Does it work? Does it not? And remembering that sometimes it doesn't work and it creates a lethal allele, and it ends it right. Nope, that didn't work. Okay. So then we have inversion. So this is rearranged. I'm gonna just leave it here. Rearranged, reinserted, and reverse. Okay. So let's say we have our chromosome, and I want to show you here because the centromere. So we could have it paracentric, so it doesn't impact the centromere. So right here, we could just do like inversion, C, B, A. But if it's, that's a paracentric. So if it doesn't involve the centromere. Now if it does, it's a pericentric. So let's look at it. We can just do it right here. E, F, D, C, right? And so it can create, there's generally a balance, right? Because you just inverted it. That's with inversion, right? We just reinserted it. So you don't really lose any genetic information. So what you could think of with this one is like a chapter book. You rip out a chapter and you stick it for earlier in the book. So it changes the plot of the book, but it doesn't really, you still have the book, right? Might change it, but it doesn't really create so much damage that you can't get through the story, right? Okay. Now, the last one is translocation. Let's just go ahead and leave this up just in case. Translocation is the double strand breaks. So there's a break, and then it's relocated. So AB, that broke off. And then it comes over here, AB. Okay? Double strand breaks, faulty repair, right? So there's a faulty repair. So there's a break, it broke off and it repairs it, sticks it on maybe a different chromosome. Maybe there is a faulty crossing over. So when we talked about that genetic recombination, maybe it crosses over unsuccessfully, right? So maybe you're helping this friend write this book. You have this chapter and you send it to the wrong address, right? So you have this chapter and you sent it to the wrong address. So you sent it to another chromosome or something, right? And so that can create problems, right? Because then you are missing these genes and it's like, wait, well, I never got the chapter, so I don't know what's supposed to happen right here. Okay? We're missing the ending because you sent that chapter somewhere else. Okay? So we talked about deletion, we're missing. So you don't have that genetic information, so you're missing chunks. So the most common one that you can think of is Cri-du-chat. Duplication is repeated. There is unequal crossing over, but there is going to be a increase in dosage of a certain gene, right? And then a deletion of another one because that loop is created. Inversion is just rearranged. So maybe you're flipping the chapter in the book. You decided, I'm going to put this chapter over here. So you can see that sometimes it works and sometimes it doesn't, but it's nature's way of kind of experimenting with different genes, or else we would have one copy and another copy and another copy of me, just clones of each other, right? So this is nature's way of getting diversity within the gene pool. However, sometimes it doesn't really work too well. And other times it's like, yeah, for some reason, sickle cell, they're resistant to malaria, so it works. So that's not a chromosomal, that's not a chromosomal mutation. That's actually a genetic mutation. But you get what I'm saying. Like sometimes the mutations, they work, and sometimes they don't. Okay. All right. So we covered a lot of information. You guys let me know if you have any questions, and thank you guys for listening.